Hello. Welcome to a brief guide to shotgun metagenomic sequence data analysis. My name is Dr. Nastassia Patin, and I'm a postdoc with the Atlantic and Oceanographic Meteorological Laboratory, which is part of the National Oceanic and Atmospheric Administration. And today I'm going to walk you through the basics of analyzing shotgun metagenomes, specifically short-read metagenomes. So this will be largely tailored to Illumina sequence data, although it could apply to other types of short-read data as well. And you may or may not be interested in every single one of these steps. I'm not going to go into great detail on any single one of them, but hopefully this will be a good primer for those of you looking to get started with metagenomics, and then you can take this information and build on it as you see fit. So this will be the general workflow that we'll follow, and don't worry if this looks a little complicated or daunting. We're going to go through every step. So the basic sequence of events will be to take your raw short reads. Again, I'll use an example of paired end Illumina data because that's the most common method of sequencing metagenomes today. You're going to want to do some quality control on those reads and then assemble them into longer sequences known as contigs. Your contigs can be used in a variety of ways. You can use them to identify open reading frames or ORFs. These are basically protein coding genes. Or you can also bin your contigs into metagenome assembled genomes, or draft microbial genomes called MAGs. You can annotate both your contigs, your ORFs, and your MAGs using a variety of databases. And finally, you can take your short reads and map them back, align them with your longer assembled contigs, and get a sense of coverage, which will give you an idea of proportional representation or relative abundance of your contig in your metagenome. So let's focus on quality control first. A lot of metabarcoding or marker gene workflows, if you're familiar with those, will include read quality control in the workflow that you use. But this is not the case for shotgun metagenomics, and so you're going to want to do your own quality control first. I like to use a program called FastP. This is a program that does adapter detection and filters reads based on quality score using the sliding window model. So rather than just looking at every base individually, it will take a window of whatever size you define and then take the average quality of the bases within that window, slide it along the sequence, and if the average drops below a certain level, those reads will be filtered out. FastP does occasionally miss adapters, which can sometimes be left on the raw reads, even though Illumina machines are supposed to trim them off. And so for that, you might want to use something called FAQCS, which is very good at adapter detection. As an example, you can visualize your reads in this program called FastQC. And what you're looking at here is a quality, average quality of reads based on position, or nucleotide position in the read. So starting at position 0 all the way to 151, which is the typical length of an Illumina short read. And you can see that towards the end of the sequence, the average quality drops quite a bit. And this is pretty common for any type of sequencing. And so if you look at your reads after having run them through FastP, you can see that this average quality has gone up, which is what you want. You've basically gotten rid of the most egregious low quality reads. You can also see in your raw forward reads that there are still some adapters left on the sequence. That's what the black line here is showing. And then after running them through FastP, you can see that those adapters were removed, no longer exist. So that's the kind of thing you want to see in your QC processing. So once you have your high quality short reads, you can do a number of read-based analyses. So this is without assembling them into longer contigs. You can take just those raw reads and do a variety of things. This is helpful because a lot of reads will fail to assemble. They'll be left out of your assembly essentially, and so you've lost that information. And so some analyses that rely on using just short reads include some taxonomic composition analyses. You can use programs like Kraken2, PhyloFlash, SingleM. You can also just blast your reads against any database. The NCBI and our database will be the most comprehensive. And then you can run something called Megan on your blast results, and that will give you an overview of taxonomy just based on your BLAST annotation to NCBI. You might also want to normalize your short reads. This often will improve your assembly quality. This is especially true if you're having trouble with your assemblies. And the most common and I believe best way of normalizing is a program called BBNorm, produced by the JGI, Joint Genome Institute. And this will basically, if you have reads that are highly overrepresented or underrepresented in your metagenome, it will sort of even out those representations, and this will very often improve your assembly quality. You can also do analyses based on k-mer composition. I'll get into a little more detail on k-mers in a little bit, but this can basically be used as a proxy for community composition. And so my favorite program of doing this is called MASH. This is a min-hash method for taking k-mers without any annotation. So you're not matching, you're not using any databases or matching to anything, and you're just assessing the composition of your metagenome based entirely on k-mers. And finally, you can look at the coverage of your metagenome. So how deeply did your community get sequenced with your metagenome using something called NOMPAREY. NOMPAREY is a program produced by the Costas Lab at Georgia Tech, and it shows you essentially the coverage of your metagenome based on sequencing effort, and then we'll extrapolate that information to show you how much sequencing you might have to do to fully sequence your sample. So in this example, you can see that these curves represent sequencing effort on the x-axis against estimated average coverage on the y-axis. And in this example, I have two kind of groups of metagenomes, and you can see that the blue group has better coverage on average because the slope of this line is steeper, and you're going to get to your fully coveraged metagenome with less sequencing effort. So I highly recommend doing something like NOMPAREY if you are concerned about the depth of your sequencing and what it might represent. So getting back to this k-mer sequence composition that I mentioned earlier, what does that mean? What is that all about? And this becomes important for several analyses down the line as well. So early on in research into microbial genomes, researchers found that you could separate those genomes into different dinucleotide composition space. So essentially, different genomes representing different taxa have corresponding composition of dinucleotides. So what does that mean? How do we compute a dinucleotide or k-mer frequency? So in this example, we have a short sequence, and what we can do is go run basically a sliding window formed of two nucleotides along your sequence and tally up how many of each dinucleotides exist in this sequence. So if we go all the way along this particular sequence, you can see it has a certain proportion of dinucleotides represented. If you do this for multiple sequences, what you find is that sequences that are more closely related phylogenetically will also have more similar dinucleotide frequencies. And you can actually cluster those and visualize that clustering in some sort of ordination. And it can be a proxy for taxonomic similarity without knowing anything about the annotation of your sequence. So when k equals 4, we call this tetranucleotide frequency, and this is the most common frequency used to classify metagenomes or assess these metagenomes for a taxonomic proxy. Okay, so that's just a quick rundown of some read-based analyses you can do. Now let's get into the heart of metagenomes, which will be assembling your reads into longer sequences called contigs. So assembly, the general principle of assembly is to find overlapping short reads to make longer ones. But the devil's in the details here. There are a couple ways of doing this. Here are some of the most common assembly programs. My favorite is called Spades, but many people prefer MegaHit, which is much faster. IDBAUD is another common assembler. And the other three listed here, I'm not very familiar with, so I'm not going to talk about them, but There are plenty of other options for assembling microbial metagenomes, but most of them will use what are called de Bruijn graphs to assemble contigs. So if you see that term, it generally refers to metagenome assembly. And what is a de Bruijn graph exactly? So it's kind of an improvement over the original overlap layout consensus assembly method, which was the first most basic way of doing assembly. And OLC is exactly what it sounds like. You take reads that overlap with each other, and then the most prevalent sequence or consensus sequence will be your contig at the end of the process. De Bruijn graphs are a little bit different. They build graphs with branches, and so instead of just eliminating your divergent sequences, it will actually graph them out, as you can see here, and show where they diverge, and then show where they reconverge. And then you can take those graphs and use them in various ways. In this particular example, the final output is going to be the same as OLC, but if you can imagine, sometimes these graphs have many different branches, and it is not just the consensus sequence that becomes your contig. I'm not going to go into more details on the computational method for this. I encourage you to look into it if you're interested. But de Bruijn graphs are really the major way we approach assembling metagenomes today. So once you've assembled your reads into longer contigs, then you can start to do some more interesting things with them. The first thing you might want to do is identify open reading frames or protein coding sequences. So this process of gene prediction is usually done by defining a sequence by its start and stop codon. But there are a couple different definitions for what an open reading frame is. It may be a start and stop codon. It may also be the end of the previous stop codon to the end of the next stop codon. That's actually the more common way of defining ORFs now. And so it's worth looking into how exactly your ORFs are being defined, and which program you might want to use based on your preference. Prodigal is the most common ORF prediction program, and it's very, very good. But there are newer or slightly different programs coming out, including machine learning based program that I don't have any experience with, but would be interested to try. And you want to think about the outputs that you're going to be working with, and what you want those to look like. And so at a minimum, you do want your nucleotide and amino acid outputs so that you can do both nucleotide and protein based searches in your annotation process. So once you have your genes identified, you probably want to know what they are encoding. And so this is a process called annotation. And annotation is just assigning your likely biochemical function to the identified open reading frames. When you have large metagenomes, identifying every single ORF on every single contig can be very computationally intensive. And so to save time and memory, you probably want to cluster your ORFs at 90% identity, which is the standard threshold for nucleotide sequences, or 70% identity if you're using amino acid sequences. Then you can extract the longest sequence from each of your clusters and run just those representative extracted sequences against your database of choice. And so 2 examples of clustering are shown here. CD-HIT is an older and very common way of clustering sequences. It tends to be quite slow, and so newer programs, including something called MeshClust, have shown improvements in speed and efficiency. They do cluster slightly differently, though, which may or may not change your final output. The program I like to use is actually called LinClust. I'm not showing that here, but it's very fast and very good at clustering sequences. So then you take your representative longest sequence from your clusters and run them against your database of choice. This is a really vast and complicated part of the pipeline, and I'm not going to go into deep detail. I will divide it into two goals. If you want just a quick functional overview, you have a couple options. But if you really want to dig deep and be extremely thorough and extensive in your annotation, you have a couple more options that will take longer, but give you more information. So for your quick functional overview, there's a program called Proca that is a wrapper combining Prodigal, so it actually will do your ORF identification for you, RNA Emmer, which is will identify RNA sequences, BLAST plus, and HMMER3 against a set of core and HMM databases. I'm not going to talk about hidden Markov model or HMMs today, but I encourage you to look into them. They are a really valuable tool in annotation and in other bioinformatic applications as well. Another great program for fairly quick annotation is CoFamScan. This is a standalone tool to search against HMM profiles of KEGG orthologs. So KEGG will be your main database used there. I think Proca uses Uniprot as its main database, but I'm not 100% certain about that. If you do want to do a more extensive or customized annotation process, you might want to use something called BLAST or Diamond. Diamond is only for protein sequences, but it is much, much faster than BLAST. And so if your metagenomes are enormous, then you might want to consider Diamond searching your protein amino acid ORFs. And you can run those against NCBI, which will be the biggest and most extensive database. Uniprot, also very large. Swissprot is the manually curated version of Uniprot, so you'll have higher confidence in your annotations, but they won't be as extensive. CASI is for carbohydrate related enzymes, and there are several others that you could look into. And finally, I want to mention a fairly new program called DRAM. DRAM stands for Distilling and Refining Annotations of Metabolism, and this is another wrapper that will use multiple databases and output your results in some very nice tables and heat maps, as well as give you the raw spreadsheets so you can do your own visualizations or analyses if you want. DRAM can be installed as a standalone program, or you can run it on the K-base server. And I've had some really good experiences with DRAM and look forward to using it more. The one caveat is I'm not sure it can be used on shotgun metagenomes right now. I used it for genomes, and so I'd have to take a look and see if you could input an entire set of metagenomic contigs. So what more can we do with our contigs and our genes? We can calculate their representation or relative abundance in the community using coverage values. So coverage is calculated by aligning, also called mapping, your original short reads onto your assembled contigs. And the most common identity threshold for your alignment will be 95%, but you might want to play around with that or customize that based on your purposes. Alignment files are going to come out in BAM or SAM format, and you're going to need to manipulate those further for any sort of more basic spreadsheet output. So the most common mappers will be Bowtie2, Minimap2, which is much faster than Bowtie and very, very good, or something called Magic Blast, which is really highly customizable. So Bowtie2 and Minimap2 will have your identity threshold set, I believe at 95%, whereas Magic Blast you can really customize, as you would for any other blast command, you can customize all the inputs and outputs, and it will give you the output in a table format, a blast output table, rather than SAM or BAM files. So you may want to use Magic Blast if you want to do some comparisons with different alignment thresholds, or you just don't want to deal with SAM or BAM files. So just to go over the principles a little bit here, coverage, again, it's the average number of short reads mapping to each nucleotide position in a contig. And so when you compare those average numbers, you're going to get different levels of coverage. And these differences in coverage can help you differentiate microbial populations or just gene frequencies in a metagenome. So that's a little preview into the next step of the workflow, which will be binning your contigs into metagenome assembled genomes, or MAGs. So using k-mer composition, which I talked about earlier, and coverage, we can group contigs into likely genomes and then string them together into metagenome assembled genomes, or MAGs. So this is a graphical representation of what I mean, where tetranucleotide frequency is on the x-axis and coverage in a metagenome is on the y-axis. And so your contigs with both similar k-mer composition and similar coverage values can be grouped together and defined as likely MAGs, basically belonging to the same microbial population. You can also leverage different coverage levels across multiple samples if you have that. And there are some binning programs that can take multiple samples and use that information. So for example, if you have some contigs that have, coverage of 1,000 in one sample and coverage of 100 in another sample, that proportion should be the same. If it's representing the same organism, it should be the same for all of your samples. And so then you can group those contigs together as likely similar organisms or populations. So here are some common binning programs. I like MaxBin2. Metabot2, also very common. Group M, Concoct, and Binsanity, I don't have any personal experience with, but I think can be good. And finally, something that I've been using recently is called VAM. This is a machine learning based binning program. And what I've found with VAM is that it doesn't necessarily give more or better MAGs than MaxBin2, which is what I compare it against, but it does give different ones. And so I've been using a combination of binning programs, generally MaxBin2 and VAM, to thoroughly bin my metagenomes. And I would encourage you to do the same because every binning program works slightly differently and will give you slightly different output, but there's no consensus on which is better. And in fact, I would say that their efficiency or their quality will depend on the type of samples you have, the type of coverage in your metagenome. All these things can affect binning efficacy. And so you should really run more than one binning program to get the best results. Finally, I want to mention something about how mapping identity threshold can influence your MAG coverage. So I talked a little bit about this earlier, how if you set your alignment threshold at 95%, you're going to get, a set of reads that map at or above that level, and that's all you're going to get. This plot is it has a lot of information. I just want to focus on this top part here, where you can see that if you go along the position in your metagenome or in your genome sequence, and then look at average depth across that genome, you're going to get essentially a histogram or a histogram of that coverage over on this panel here. And you can see this is one very sharp peak, well defined. Now, if you look at this example, you can see this top peak is accompanied by a secondary peak where another set of reads has been mapped at a lower depth of coverage. And if you look at that mapping now in this bottom panel here, which shows position by the percent identity of each read, you can see that there's kind of a secondary cluster at a lower percent identity that's mapping to your genome sequence. And what that suggests is that there might be two populations that are closely related, but not identical. And you can see that in the different levels of coverage that each of them have. So just something to think about when you're doing your mapping and thinking about differentiating microbial populations and MAGs and what they mean. So once you have your MAGs, you should really only work with high quality MAGs because posting low quality, incomplete genomes onto public databases can be a disservice to the community. Someone might download a genome and think it represents an organism, but in fact it's highly contaminated. Contamination can also be called redundancy. CheckM is sort of the original and still most standard way of checking quality of your MAGs, and it uses the term contamination to refer to multiple copies of what should be single copy genes. Onvio is another program that does a variety of things. I'll talk a little bit more about it later, but it refers to this as redundancy because there's no hard proof to show that there's any contamination in this genome. It may be actual biological redundancy. But in any case, they both work on the on the presence of single copy core genes that should be present in every single um, microbe, and it has different sets of genes for bacteria and archaea. Next, you want to assign taxonomy to your genomes, your high quality genomes, and for that you should use something called GTDB-TK. GTDB is the Genome Taxonomy Database that uses a phylogenetic approach to assign taxonomy. And you can run this as a standalone tool. You can also run it, you can use Onvio to run your MAGs against the database itself, but this analysis is based on ribosomal protein sequences. It's not a phylogenetic approach like GTDB-TK. And so I've had many instances where my MAGs will fail to be annotated by Onvio, even though it's using GTDB. But if I run it through GTDB-TK, I will get the closest relative and likely annotation, taxonomic annotation. So just keep that in mind if you want to use Onvio for your taxonomic assignment. Then you want to make sure, if you're working with multiple metagenomes, you might get many MAGs that are annotated as being closely related or the same population. And in the next stages of your analysis, you don't want to be working with duplicated genomes. That's just silly. So you want to dereplicate your MAGs. And the best way of doing that is with a metric called average nucleotide identity, or ANI. And ANI is calculated for all orthologous genes that are shared between two genomes. So it calculates first how many genes are shared, and then, based on that shared fraction, what is the average sequence identity. And generally 95% genome-wide ANI, or GANI, is considered a species-level delineation for microbes. But if your alignment fraction, so the number of genes that are shared between your two genomes, is under 70%, ANI will not be a useful metric, and you should turn to average amino acid identity instead. And so on the right, you can see some charts showing how sequenced genomes compare to each other using ANI. And this, there was a fair bit of work done on this in the last 10 or so years, and it turns out that the 95% species level definition came from these meta-analyses that showed a clear separation in groups of genomes. This might be shown a little better on these graphs, where you can see that many genomes are between 95 and 100% ANI. These are all very closely related, likely species, same species, if that's how we're going to define them. And then there tends to be a gap. And then you get another group of genomes that are related at between 75 and 85% ANI. And that might be the nearest neighbors within the genus or the family. These analyses were done on over 90,000 prokaryotic genomes. And so it does seem to be a biological principle that this genetic discontinuity is based on ANI. And that discontinuity holds up with MAGs as well as with sequenced genomes. So some dereplication programs include DREP, FastANI, and PyANI. And they all work in fairly similar ways. DREP has an initial deconvolution step just based on Kamer composition using MASH. And so that can work a bit faster. But they all use ANI to do the final delineation of your MAGs. So that's a lot of steps. And wouldn't it be nice if we could just feed in our raw reads and get all the outputs that I mentioned in this workflow spit out with minimal effort? Unfortunately, or maybe fortunately, depending on your perspective, that's not really, it's not common right now. there are certainly wrappers that exist. I have limited experience with them, and I would recommend doing all of these steps manually if you're just getting started, so you have an understanding of the process. But there are a couple options if you want to automate parts of this workflow down the line. Onvio is a very common and very powerful pipeline that works post-assembly. So once you have your reads assembled into contigs, you can feed those into Onvio and run a series of steps that will get you through the binning and annotation process. I don't like to bin with Onvio either, personally, but it does give you that option if you choose. Atlas and this other one called Stag I'm not familiar with them, but I believe they're snake-make pipelines that will run through assembly all the way through binning. And then DOS-Tool is a pipeline it's not really a metagenomic pipeline. It's only for binning, but it will run multiple, or it will take outputs from multiple binning programs and dereplicate and score your bins and give you the best, highest quality output from multiple binning programs. So it's very, very useful to assess your binning results, but that's all it does. It doesn't do any other part of the workflow. So if you're just getting started, again, I encourage you to do every step manually. And then this is a really exciting field of research right now, and I have no doubt that there will be more efficient, more automated pipelines that will still allow for some customization down the line. So you don't just need to follow, the script provided by the pipeline. But for now, most bioinformaticians, people interested in metagenomes, will do most of these steps manually. And so finally, I just want to leave you with a bit of a thought exercise when we're talking about MAGs and binning out genomes from metagenomes. It sounds really, really simple, really great, and we often think of different microbial taxa as being these sort of distinct genomes floating around in the environment. But the truth is that microbes are constantly exchanging genes by horizontal gene transfer, and they're constantly evolving vertically as well. And often you might have a group of very closely related populations where microdiversity is so high, you can't really get a consensus genome. And yet you want to find a way to represent these populations and to understand how their taxonomy and their function are linked. And it's a real challenge. I work in marine systems, and the most ubiquitous microbe in the ocean, SAR11, is actually one of these organisms where there is just so much microdiversity. It's very, very challenging to bin out a MAG. In fact, I've never really been able to do it, not a high quality one. And so what are the opportunities, or sorry, what are the options we have then? If regular binning doesn't really get us to where we want to get with a lot of these microbes. And this paper came out recently that talked about using pan-metagenomes rather than trying to bin population level MAGs. And the title of the paper is Embracing Metagenomic Complexity with a Genome-Free Approach. And the chart on the right kind of shows how closely related microdiverse populations can be really problematic for assembly and for binning. And even if you do get a bin, it's likely leaving out a lot of important information, a lot of important genes that are prevalent in this taxon, but maybe not at the threshold level that you've defined for a MAG. And so maybe we should be thinking about these populations in terms of pan-metagenomes rather than sort of linear consensus MAGs. I think it's a really interesting concept, and I think it's going to be a really important concept as we continue to explore and define microbial diversity in the environment. But there's no program or algorithm yet that is getting us there. And so right now it's more of a thought exercise, a theoretical concept. But I encourage you to think about this when you're binning your MAGs and and make sure not to misrepresent the product of those analyses. So with that, I will end this tutorial. I hope it was useful for you. And please feel free, I'll put my contact information in the comments or in the description here. Feel free to contact me with any questions. Thank you.